Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and what we were discussing about the protein production strategies in different types of host. In this module, what we have discussed so far, we have discussed about the protein production strategies in the E. coli as a system, then subsequently we have discussed about the yeast as an host, which is actually the basic eukaryotic host and unicellular eukaryotic host. So, the manipulation as well as the protein production in the eukaryotic system such as yeast is the simplest system. Then subsequently we have discussed about what is the disadvantages of using the yeast as an expression system, and then subsequently we have talked about the insect cell line as a expression system. In that we have discussed about how to clone the proteins into the baculo expression systems, how to screen them, how to select, and then ultimately how to generate the recombinant viruses which you can use to infect the insect cells, so that the insect cells will overexpress the proteins. And at the end we have also discussed about how to retrieve these cells, whether the cells would be cytosolic in nature or the protein what you are going to express in the going to secrete into the media of the insect cells, and both of these strategies have the different requirement in terms of the media what you are going to use for protein production. So, now continuing the discussion about the different types of host, today we are going to discuss about the mammalian cells as an expression system. So, mammalian cells, there are different types of mammalian cells which are being used for protein productions. So, mammalian expression system is the most complicated eukaryotic expression system which people are using for protein production, as well as for asking the questions or studying the transcription as well as the translation of the foreign gene or foreign proteins. Sometimes they are also looking for what will be the impact of or what will be the functional role of a particular protein. In those cases they overexpress that particular protein into a cell and then ask what will be the physiological as well as the phenotypic changes occurs in that cell. So, similar to all other expression systems, the protein production in the mammalian cell system can be achieved with either from a vector based such as the extra chromosomal DNA, and once you produce from the extra chromosomal DNA, it is going to be transient, because the extra chromosomal DNA is going to be degrade in due course of time, or some cases you can actually sequence you can integrate into the genome through homologous recombinations to establish the permanent cell line. So, the expression from the permanent cell line will be intact, it will going to express as long as you grow these cells. The expression from a transient or the permanent cell line can be from a constitutive as well as the inducible promoter. As we discussed in the yeast, as in the case of yeast, you have the choices of either using the constitutive promoters or the inducible promoters in the case of mammalian cells as well. Irrespective of the expression mode in mammalian system, the different basic steps which you are required to produce the protein are as follows. The first step is that you have to clone the foreign gene in mammalian expression vector. We in our previous lecture we have discussed different types of mammalian expression vector which are available for cloning the foreign gene into the vector. Then once you generated the foreign this recombinant clone, then the second step is you have to transfect the cell line with the recombinant construct. This one also, the transfection of the mammalian cells also we have discussed in detail, where you can use either the chemical method or the lipofectamine, or there are 3 to 4 different approaches what we have discussed in the case of mammalian cells and the transfection of the mammalian cells. Then once you transfected the cells, you have to screen and select the transfected cells, and ultimately you have to culture these transfected cells, and then you have to do the protein production. So, let us continue with the mammalian expression system and understand all these steps. So, In the mammalian expression system, you can use different types of cell lines for protein production. These are not the exhaustive list of cell line what you can use, but these are the different cell line which are very popular in terms of protein production as well as performing the experiments into the labs. So, you have the different types of cell line and they are being derived from the different origins. For example, you have the MCF7 which is a breast cancer cell line, you have the HT1080, you have EH927 which is a endothelial cell line, and then you have the J774A.1 which is a macro cell line. I have given the list of cell line here also, in which you have the CV1 cell line which is actually from the kidney. Then you have the COS7 cell line which is from the fibroblast origin. Then you have the NIHTT3 which is a fibroblast. Then you have the CHOK1 which is actually the ovarian origin. Then you have the J774A.1 which is of the macrophage origin, HeLa which is a cervical cancer cell lines, then BHK21 which is a kidney cell line, and HEK293 which is a kidney cell line. And among these cell line, why we are talking about this cell line, because you have to choose this cell line as according to the origin of the foreign gene. For example, if I have to overexpress cytokine gene into the for protein production, I would prefer to use the immune cells. In those cases, I may prefer to use the J774 which is a macrophage cell line. Similarly, if I have a protein production, I would like to just use some mammalian cells, then I will go for those cell line which are easy to propagate, easy to grow, and the manipulation of these cells in the cell culture would be easy. In those contexts, you can use the either the COS7 cells or the CHO cells, because these cells are very easy to grow in the mammalian into the mammalian media, and these cells are easy to manipulate compared to all other cells which the other third cell line what you can also use is HEK293 which is actually very easy to manipulate, very easy to transfect, and very easy to propagate within the media. As we discussed, you can have the two different mode in which you can get the protein expression. Either it would be a transient expression or it would be a permanent expression. So, transient expression would be the expression where the expression is going to be very high, but it would be a very short period, which means if you transfect the cells with the DNA expressing the protein, it will going to give you the protein production until the 72 after the post transfection. After 72 hours, the extrasomal DNA or extrachromosomal construct which you are going to transfect into the cell is going to be degraded by the cellular machinery, and eventually the protein production is going to stop. Transient expression system is used to screen the cDNA libraries, isolation of a particular cDNA clone expressing surface antigen, or to test the applicability of the recombinant construct going to be used for permanent expression. So, before you go for the exhaustive exercise of permanent expressions, you also want to test whether the construct what you have prepared is going to express and give you the protein or not. For those kind of expression as well, you always use the transient expression, but in general the transient expression system is not being used very extensively for protein production. It is always being used to answer the research related questions. For example, if I overexpress this particular protein, how and what way it is going to change the morphology as well as the phenotype of that particular cell. For those kind of experiments, you use the transient expression. But if you want to do the protein production, the transient expression system is going to be very costly, because you are not going to get the protein production post 72 hours. This means after every 72 hours you have to do another round of transfection, which means you have to isolate the DNA, you have to make the supercoiled high quality DNA which will going to transfect into the mammalian cells, and then you also have to use the transfection reagent. These transfection reagents are going to be costly. So, if you do multiple rounds of transfections and try to use those cells for protein production, it is not going to be economically viable options for protein production. For that purpose, people always used to prefer making the permanent transfections or permanent expression system. So, in the permanent expression system, the permanent expression system you are going to use, and you will use the gene and integrate into the chromosomal DNA. Once you integrate that into the chromosomal DNA, the protein will going to be expressed along with the, or the number of protein gene, or the once as soon as the cell will divide, you are going to have the more number of copies of that particular gene. So, it is actually going to continue along with the cell division. So, that is why even if the cells will divide, it is, you are not going to lose the DNA, and that is why it is called as the permanent expressions. The most crucial step in the permanent transfection is the establishment of permanent expression system for a gene is to integration of that particular gene, and that it depends on the number of times or number of frequency of integration into the genome. So, you have to achieve that by simply screening the cells on multiple rounds, just to see that it is getting integrated into the genome, and that is why the permanent expression system or development of the cells which have the permanent expression is a time consuming as well as the labor intensive exercise. But once you are done, you are sure that this particular protein is going to be expressed from these cells lifelong. You do not need to do another round of transfection or selections. And so, initial steps are time consuming, but subsequently it is going to pay you in terms of reducing the cost of the transfection reagents, or making the DNA, as well as it is going to reduce the manpower requirement to do these jobs. In simpler words, the permanent transfections depends on the recombination frequency instead of transfection efficiency. So, let us start with the transient expression and understand what are the different steps you have to perform to make the protein under the transient expression systems. So, as I said, the transient expression system, the expression is high, but it is short lived. The cells transfected with DNA express protein until 72 post transfections. Transient expression system is used to screen cDNA libraries, isolation of a particular cDNA clone expressing surface antigen, or to test the applicability of the recombinant construct going to use for permanent expressions. There are multiple steps required to transiently express a protein in COS7 cell lines. So, in this particular example, we have taken the COS7 as a COS7 cell line to show you that how to do the transient expressions, but for other cell line also the steps are going to be remain constant, except that you might have to optimize or standardize these steps depending on what cell line you are using. Suppose some cell line you are using which are not adherent cell line, non-adherent cell line, then you have to change little bit, but more or less the steps are going to be remain constant. So, the procedure can be applied to other cell line, but with slight modification. The steps which you are going to use for making a transient expression are as follows. So, in the first step, what you are going to do is you first plate the COS7 cells into a plate, okay, and then what you have to do is the first step is you take the supercoiled high quality DNA and do the transfections. So, first is the clone the foreign DNA into the appropriate mammalian expression vector to obtain the recombinant DNA, and then the as we discussed in the past also, the transfection efficiency is maximum for a supercoiled circular DNA. So, you have to purify the recombinant DNA by a mini prep to prepare the high quality supercoiled DNA. Once that is done, then the second step you have to do the transfection, and once you are done with the transfection, so in the second step what you do is you seed the COS7 cells in DMM media, which is actually the standard media recommended for growing the COS7 cells at a confluency of 20% in 100 mm dish. Once the cells are adhered and they are ready, then you do a transfection of these cells with the recombinant DNA which you are going to use with the transfection reagent. You have the option to use the different types of transfection reagent which we have discussed in the past. You can also use the virus mediated or the bacteria mediated DNA delivery as well. So, these are the different methods. Once you are done the transfection, these cells are now being transfected. Then what you have to do is, since all of these recombinant DNA is going to have the selection pressure, you have to select these cells. So, that is the third step. You have to select these cells, and suppose in this case we have taken an antibiotic. So, what you have to do is you grow these cells in the presence of antibiotics. As soon as you put the antibiotics, the most of the cells which are not being transfected are going to die, and then you are going to have the cells which are being transiently transfected. So, these cells are expressing your protein, and along with that these cells are also expressing the antibiotic resistance genes as well. Now, what you have to do is these cells are ready to use for transient expressions. In the transient expression, you have the option whether you want to have the protein onto the surface or the cytosol, or you want to protein to be expressed or secreted out into the media. Depending on the conditions, you can use the different types of media. So, imagine that if the protein is present on the cell surface or the intracellular protein, which means that the protein is associated with the cellular fractions, then in that case you so transfected cells are allowed to express the protein for another 72 hours, remove media, and then wash the cells with PBS and detect the presence of protein on cell surface or in cell lysate by activity assay or the Western blotting. So, once you have these transfected cells, you centrifuge them. That will give you the cellular component as well as the supernatant, which is actually the media. The cellular component you can lyse, or suppose the cells are present on the cell surface, then you can take these cells and use the FACS to detect the presence of antigen on the cell surface. Otherwise, you can lyse these cells and detect the protein into the intracellular or cell surface protein using the Western blotting or the activity assay. But if the protein is expressed and secreted into the external media, then what you have to do is, if the protein is expressing secreted out, then you wash the cells with PBS and add serum free media. Okay. It will allow to secrete the protein for another 72 hours. Then what you do is harvest the media, remove the dead cells and debris by centrifugation, and filter the media by a 0.45 micron meter. So, if the cells, if you are putting the protein into the secretory pathway, the protein will present into the supernatant. In that case, what you do is you wash these cells with the serum free media and then incubate them into the serum free media. These cells then will secrete the protein into the supernatant. You centrifuge, you recover the cells, you centrifuge, and that will give you the cellular component as well as the supernatant. This supernatant you can pass through to the 0.4 micron meter, so that you can remove all the dead and damaged cells, and you will only going to get the clear supernatant. Once you have done that, then you can detect the presence of your protein in the media either by activity assay or by Western blotting. So, then you have the secretory protein and you can do the Western blotting or the activity assay for presence of your protein. So, this is all about the expressing the protein into the mammalian expression system under the transient expression. But if suppose you want to make the permanent expression system, then you have to first generate the cells under the permanent transfectant, and then once your cells are ready, then you can use them for protein production. So, under the permanent expression, the permanent expression is of a gene is possible if it will be integrated into the chromosomal DNA. The most crucial step to establish a permanent expression system for a gene is the frequency of integration event rather than the number of DNA uptake. In simpler words, the permanent transfection depend on the recombination frequency instead of the transfection efficiency. The stable transformants are selected by a selection marker either as an antibiotic or the auxotrophic factor or a negative selection with an inhibitor. All these the selection of the cells we have discussed in the past, where either you use the antibiotic, which is going to be a positive selection, or the auxotrophic method, where the media is not going to have the particular type of nutrients. So, the cell will not be able to grow, but once your recombinant DNA will be there, that recombinant DNA is going to provide the nutrients, and that you can use even on the nutrient deficient media. And the third is the negative selection, where you are going to add the chemicals, and that chemical is going to kill the cells, but if the protein is the chemical is being inactivated by the presence of your particular recombinant DNA, then the cell will survive. But the question is, this screening has to be done for a prolonged period of time to ensure that the recombinant DNA is getting integrated. You have the different steps which you have to follow for making the permanent expressions. What are the different steps? So, in the step 1, you first clone the foreign DNA into the appropriate expression vector to obtain the recombinant DNA. This step is exactly the same as we have discussed for the transient expressions. You know that the transfection efficiency is going to be maximum for supercoiled circular DNA. Then you purify the recombinant DNA either by the mini prep or in a high quality supercoiled DNA. Then what you have to do is seed the cells in DMM media supplemented with 10% FBS at a 20% confluency in 100 mm dish. Then the third step, you have to do a transfection. So, transfect these cells with a recombinant DNA using a transfection reagent. This step number 1, 2 and 3 are going to be the identical what we have just discussed for the transient expression system. Now, in the step number 4, you have to do a selection of the transfected cells, and once you have selected the transfected cell, then you can take a small aliquot of these cells and test the expression of foreign protein with the Western blotting. In addition, since we are looking for the permanent expression, you also have to see that the gene is getting integrated into the genome. That you can check simply by performing a Southern blotting with a radioactive probe derived from the gene of interest. If you remember, we have discussed in the past how to generate, how to prepare the radioactive probes. Either you can use different types of method or different types of approach which we have discussed while we were discussing about screening of genomic library as well as the cDNA library. So, the similar kind of approach you can use to prepare the radioactive probe of the sequence which is from the foreign gene, and then that you can use in a standard Southern blotting techniques to detect the genome, and if it is going to give you the signal, that will indicate that your gene of interest is being integrated into the genome. So, these are the 5 steps you are supposed to do. We are not going to discuss about step number 1, 2 and 3, because these are we have already discussed in the past in terms of the transient expression. We are going to discuss about the step number 4 about the selection of transfected cells in the case of permanent expression. So, in the case of permanent expressions, the step number 1 is that you first plate the cells at a 20% confluency. Step number 2, you do the transfection so that the cells are getting transfected. Then you are going to put them under the selection pressure, either it is antibiotic, auxotrophs or the negative selection. Once you do that, you are going to see that some of the cells are going to survive, they will not going to die, because these are the cells which are getting transfected, and these are the cells you have to select. So, in the 48 hours after the transfection, split the transfected cells in a selection media containing antibiotic and allowed to grow for another 4 days, which means once you are done the transfection, you take out these cells and plate it into another dish which contains the antibiotic and let them to grow for another 4 days. In these 4 days, the lot of cells are going to die, and what you have to do is every day you have to gently wash the cells with PBS and observe that you are going to have the discrete colonies or not, which means this you have to continue by gently washing the cells, so that all the dead and the damaged cells you should remove from the dish, otherwise these dead cells are also going to damage your transfected cell as well. Now what you have to do is, since you got these dishes, what you have to do is, a cell culture dish has a plate like thing, and on from the back side what you have to do is, you take the marker and then you encircle what are the places you have the colonies, like as I am doing now, right. So, that you have to do from the back side. So, delineate the boundaries of each colony with the marker from the back side of the plate. Now, remove the media, okay, and put the cloning rings. So, cloning rings are actually the ring like structures, and they are being, once you put them onto a particular plate, they will go and get sticks. Okay. So, if suppose this is the plate and you are going to put the cloning ring is actually going to make this as like a well. So, once you put the cloning ring, the whatever you put inside the cloning ring, suppose this is your colony what you would like to isolate, the cloning, once you put the cloning rings, and the cloning ring will make this particular area impermeable, or the media will not going to leak out. Okay. Then what you have to do is, you have to add the 100 microliter trypsin EDTA. So, into this you are going to isolate this colony simply by putting the 100 microliter of trypsin EDTA. So, you fill these cloning rings with the 100 microliter of trypsin EDTA. Once you do that, the cells which are being adhered, they will come off and present into the liquid. Then you wash the colony with the PBS and transfer this single colony into another 24 well dish and let it be growing. So, allow it to grow and become 80% confluent. Once it becomes 80% confluent, then you do another round of selections. Okay. So, then what you do is, so you have put it into 24 well dish, okay, and you allowed that to go for 80% confluency. Now, what you, once it is getting confluent, then you take out these cells and put it into the 6 well dish. So, initially you started with the 24 well dish, you can and then you transfer these cells to the 6 well dish in the presence of selection media, which means you can still have the antibiotic pressure and allow that to reach to 80% confluency. So, once it is reaching to the 6 well dish 80% confluency, you can take out from here again and let it grow into the 10 centimeter dish in the presence of the selection media, and that actually is going to give you the permanent mammalian cells. Now, whether you have the permanent mammalian cells or not, that you have to test into the subsequent step. So, in the step number 5, you take the small aliquot of these cells and test the presence of foreign protein with the Western blotting. So, what you have to do is take the small amount of cells and prepare the cell lysate, and then you do the cell lysate and use it for the Western blotting, and the cell lysate you can do, and then you do the Western blotting to check whether the foreign protein is present into that Western blotting or not. In addition, since we are talking about the permanent transfection, you have to also check the integration of this gene into the genome. The standard technique is southern blotting. So, you can prepare a probe for that foreign gene and then use that foreign gene to detect the fragments into the southern blotting. It gives you the signal that will confirm that the gene is being integrated into the genome. So, this is all about the transient or the permanent expressions, and within the mammalian expression system you have the option either to go with the constitutive expression or to the inducible expressions. So, in the constitutive expression, just like as we discussed for the yeast cells, you have the option to use the promoters of the housekeeping genes, and as a result the expression is non-inducible because it is going to use the housekeeping promoters, and the protein production would be in proportion to the number of cells which are present, and as a result the expression is going to be non-inducible. So, the protein production starts with the growth of the cell, and as a result it is proportional to the cell mass. Whereas, similar to the yeast where we were using the methanol as an inducer, you can also have the inducible expression system. So, in the inducible expression system is very useful for the expression of a toxic protein or the protein with the pleiotropic or non-specific effects, which means if you are overexpressing a toxic protein or the protein which have a very adverse effect on the cellular physiology, in those cases what you are going to do, you can use the inducible promoter system, and what will happen is once you reach to a cell mass, then you can add the inducer, and that actually will allow the cells to overexpress that particular protein. Eventually, because the protein is toxic in nature, it is going to kill the cells or it is going to adversely affect the growth of the cell, but at that moment it may not be an issue because you already have a very high cellular density of these cells, and that actually will give you very high production of the proteins. So, let us discuss about the inducible expression system. So, in the inducible expression system is useful for the expression of a toxic protein or protein with the pleiotropic or non-specific effect. The tetracycline controlled inducible system is given in this diagram. In this particular system, you have the 7 tandemly arranged TET operators, and these TET operators are being placed upstream to the minimal CMV promoter and the transcriptional activator TTA gene. In the another set, what will happen is the target gene is replaced with TTA gene in the presence of tetracycline. So, what will happen in the presence of so you grow these cells in the presence of tetracycline, the binding of TTA is blocked to the tetracycline operator, which means as long as the tetracycline is present, it will not going to allow the operator to bind to the TTA operator, tetracycline operators. Consequently, what will happen? It will cause, because your operator will not going to the tetracycline operator will not bind, that is why it is not going to drive the production of the target gene as well as the expression of TTA or the target gene. But once you remove the tetracycline, okay, so in the absence of tetracycline, the low level of TTA binds to the operator, and that actually will enhance the expression of TTA, which in turn will stimulate the further amplification of initial signal. The transcription activator TTA produced in the absence of tetracycline eventually stimulate the expression of target gene, which means once you remove the tetracycline, the low level of TTA will bind to the operator, and that actually will stimulate the production of the foreign gene as well as the TTA protein. This TTA will go and bind to the another round of operator, and since initially there will be a low level of TTA, but once the it will going to produce more amount of TTA, it is going to generate very inducible system, and that is how it is going to induce the production of protein from the target gene. So, this is called as the TET on TET off inducible promoter system or inducible expression system in the mammalian expression system. We have discussed about different types of expression system. We started with the very simple expression system such as the E. coli expression system, and we have discussed many aspects or many approaches what you can use to overexpress the tetracycline expression system. Then subsequently we moved on and discussed about the yeast as an expression system, which is actually the simplest expression system in the eukaryotic system, and then we have also discussed about the insect cell line as an expression system, and by the end of this lecture we have also discussed about the mammalian expression system, we have discussed about how to produce the transient expressions or how to generate the permanent expressions into the mammalian expression system, and at the end we have also discussed about the constitutive as well as the inducible expression system in the case of mammalian expression system. So, with this I would like to conclude our lecture here. In the subsequent lecture we are going to discuss about the recovery of the protein from the overexpressing cells, and then subsequently we are going to discuss about how to recover the product, how to purify the product, and how to characterize that particular product, because as you remember we have to use these products for downstream biotechnology applications. So, all these product has to be characterized using the standard biochemical as well as the biophysical parameter associated with these products, and once these products are being verified on these parameters, then only we can put them into the application part. And at the end of this course we are also going to discuss about the applications of these biotechnology products. And with this we would like to conclude our lecture here. Thank you.